The harder question is why did these things happen? We can talk about what we saw, that some were squishy, some weren't. But why did those things happen in the potatoes? Well, it has to do with membranes, right? Membranes. And remember, membranes are just layers, or kind of like walls. They separate things in our bodies and in cells. So we added salt. Let's go and pretend this side over here is the potato, okay? And this side over here is the outside water. Sorry, that's backwards. Totally backwards. This is the outside water, because we didn't add salt into the potato. And this is the potato. Inside potato. Okie doke. So we have the outside water where we added something like salt, which is NaCl. You don't need to know what that means, just know that it's salt. And inside the potato we just have water and a few other things. Because inside the cells, we've got organelles, we've got a bunch of other stuff. But we're just going to pretend it's water for now, so it's easy to think about. So inside the potato, we have some water. And outside the potato, we add some salt. Now notice what the salt is doing here. The salt is sticking to water molecules and holding them on one side. Okay? The salt is sticking to them. So it's sticking to 6 atoms over there, okay? So water is constantly moving back and forth across this membrane. It likes to run around a lot. So it's running back and forth, okay? But since the salt is sticking to all of the water, okay, most of the water that comes over, more water is going to start moving over here because the water is getting stuck over there. So what's going to happen is that water will move out of the potato into the surrounding water, okay, because salt is sticking to water molecules and pulls water out. Basically, the salt doesn't actually do any pulling, it just does sticking. But after it sticks, then all this water is going to start moving over. So we can pretend that it's pulling. So the salt sticks to the water molecules. It sticks to them right here. And after it's stuck there, we're going to pull some more water molecules over to that side so it's equal. Because if you count the total number of water molecules on each side, you can see that there's 6 over here, and there's 12 over here, but there's only 6 free ones. So there's 6 free water molecules over here and 6 free water molecules over here. There's 12 total, but because we stuck 6, because the salt is stuck to 6 of them, we only have 6 total. So it's equal, right? We've got 6 on each side. So when we stick to water molecules, we make it equal after that. So if I stick to water molecules on one side, I have to pull water out of the potato so it gets equal. And that's the same thing that happens to us, right? So if you need to remember this, this is the person who drank, the woman who drank too much water, right? And her cells exploded. Cells exploded. Okay? Because she drank a ton of water, and we needed to make the amount of stuff equal. So so inside of her cell, right? Inside of the cells, water was sticking to other things, right? Okay? So that means that if we have a bunch of water on the outside, that we have to pull some water in so it's equal. So we keep pulling water in and in and in and in until our cells explode. Okay? This one is normal, when everything's equal. Everything is nice and equal. And then this one is when it shrivels, which is kind of what happened to the potatoes, right? They shriveled because water went out, because you had something like salt sticking to the waters, and that pulls water out of the cell to make things equal on both sides. So you get the cell starts to shrink because all this water is leaving. So this is the salt cup. This is the salt and sugar cup. This one is the water cup, right? Water went into the potato. And a potato cells aren't going to explode like ours because they're not animals, right? So we are actually going to take a look at the plant cells, because the potato is a plant. Potato. So the potato is a plant. So if we have the water cup, the cells are going to be really turgid. All that means is they're going to be hard, right? They're going to be hard because they have a lot of water inside of them. And this is squishy, and this is super squishy. Okay? So we had 3 different cups, right? The hard one to think about is the sugar cup. So we had the water cup, and we had the potato piece. Here's the potato piece, and water is going in. In the water cup, water went into the potato to make things equal on both sides. In the salt cup, we had the potato. We had some salt on the outside sticking to waters. And so water, water left the potato. Okay, water left the potato. Because salt stuck to waters. Water left the potato because salt stuck to waters to make it equal on both sides. Water left the potato because salt stuck to waters, so we could make it equal on both sides. Now the sugar cup. What about the sugar cup? Well, the sugar is just like salt. We have the potato, okay, and we've got some sugar out here. But sugar is only going to stick to like one. Sorry, let me write this here. This is a sugar, okay? So if I draw that, that means it's a sugar. So we've got sugars in here, and they are sticking to waters, the little circles, okay? Sugar sticks to like maybe 2 or maybe 2 waters. If you look at salt, it sticks to like 6 or 7, right? It sticks to a ton. But sugar only sticks to a few molecules of water. So water left the potato to make it equal on both sides. Not as much had to leave though, because sugar only sticks to a few waters, okay? So sugars only aren't going to stick to as many waters as the salt, so we're not going to pull as much water out. So it's going to be medium squishy. So this is going to be hard because water's going in. This is going to be squishy. This is going to be super squishy. And this is going to be a little squishy. Okay? That's what happened in the potato lab. And so this is another picture. You had water coming out. This is H2O coming out. H2O came out a lot. And you had H2O going in. Okay? So how does this show that membranes are selectively permeable? Well, selectively permeable means that only certain things can get through. So if you look, the salt and sugar couldn't get through the membrane. Salt and sugar couldn't get through the membrane because they stayed on one side. They stayed on the outside of the potato. But water could move back and forth. Okay, so the membrane, right here, the membrane let water through, but not the other things. Okay? That means it's selectively permeable. Selective means you pick because you're choosy. Permeable means it has holes. Okay? So if I'm selectively permeable, I am super picky about what can get through my membrane. I don't like salt, so I keep it out. But water, sure, you can come on in, go through, back and forth, whatever you like. That is selectively permeable. It picks and chooses. So it chose let the water through, but not the salt and the chlorine. But the water went through if it wanted to. But the salt and chlorine were stuck, and the sugar too. Enzymes. So draw and label a picture of an enzyme, its substrate, and what it does to the substrate and its active site. So let's review what enzymes are. Enzymes are proteins, okay? And they break down things. So the things that enzymes break apart or put together are called the substrate, okay? And the place where where the substrate is going to go in and get reacted, right? It's going to go in over here. That's called the active site. So this is the substrate in the active site. The substrate is the thing that goes inside the enzyme and gets changed. The active site is where it goes into. Those are the first two things. Oh, and the enzyme, sorry, you should draw as a big, you can just draw a big, it's just a big molecule, big protein, right? So you can draw a bunch of circles, or you can just draw something like this, right? And you could draw a little substrate that's going to go over here and get in the active site. So draw, label, a picture of enzyme, its substrate, what it does to the substrate, and its active site. So I'll show you what you could do. I'm going to draw myself an enzyme. I'm going to make it so it has a very specific shape. And here is my little substrate. So this is the enzyme, and this is what you can do in the test. This is the substrate, and this right here is the active site, okay? And then we are going to show what happens to the substrate. So it's going to go into the enzyme, and after the substrate goes in there, okay, the substrate goes into the enzyme, and then what happens after that is the substrate is broken into two smaller pieces. So I could say, sorry, the substrate is broken into two smaller things. That's what this enzyme does, okay? So this is what you could show on the test, and you would get full credit. You drew the substrate, you drew an enzyme, you drew an active site, and you're showing what happens to the substrate when the enzyme works on it. When the enzyme squeezes the substrate and does what it's going to do, it breaks that up. So what do we use enzyme for? Well, you just saw it. They make reactions happen. We make reactions between things in our body happen. And something that makes a reaction happen is called a catalyst. So we say enzymes catalyze reactions because they are catalysts. So this enzyme is the enzyme is catalyzing the breakdown of this substrate, okay? Because after it puts the little substrate in there, a reaction is going to happen, and the substrate's going to change. But the enzyme stays the same. The liver lab review. So the liver lab showed us how enzymes work. The enzyme inside the liver is called catalase. What happened when you added hydrogen peroxide to the liver? Why did this happen? So we have to talk about what catalase does. So the first thing, what will happen when you added hydrogen peroxide to the liver? Well, we had bubbles, tons and tons of bubbles coming out from the liver right there, and the hydrogen peroxide, which is H2O2, hydrogen peroxide. So we had a bunch of bubbles come out. That's what happened when we added hydrogen peroxide. Now, why did this happen? Well, catalase is an enzyme, right? Here's what it does. Normally, hydrogen peroxide just sits there, right? If you have hydrogen peroxide in a bottle, it stays hydrogen peroxide. It just sits there. And so nothing's going to happen. There is absolutely no reaction. There's no reaction happening, okay? Everything just sits there. Nothing's going to change. We get none of this stuff. We just get H2O2 forever. Okay? That's normally. But catalase is the enzyme, right? The enzyme catalase helps the breakdown of hydrogen peroxide over here, hydrogen peroxide, into 2H2O, which is water, and O2, which is oxygen. Okay? So I took some enzyme and I added it to hydrogen peroxide, and then the reaction was catalyzed. The reaction happened. And I made water and oxygen. And the oxygen was the bubbles. Okay? So we use the enzyme to cause the hydrogen peroxide to turn into water and oxygen, because we can help it break down, because the enzyme helps that reaction happen. And we could do this over and over again, right? Because we kept using the pieces of liver with hydrogen peroxide, and we could do it for as long as we liked, because the enzyme will never run out, because it doesn't get used up. It's just a tool to help break things apart. So when we heated the enzyme, it changed how well the enzyme worked. So what happened to catalase when we cooked the liver? Well, we did denaturation. We cooked that liver in the pot of boiling water, and we took this beautiful little catalase and we broke it up. Denatured or broken catalase. We broke the catalase. If we break the enzyme, it can't do its job. So we can't turn any more hydrogen peroxide into oxygen. So in the hot, if we had the boiled liver, there's no bubbles. No bubbles. Because the enzyme is broken. The enzyme is broken. We broke it by heating it. Okay? We denaturation because of heat. We had heat and we denature that enzyme. We break it up and separate it out, and it can't work anymore. So we have no bubbles. This is the normal one, right? And it makes a lot of bubbles. If we put it on cool, cool the enzyme down, it just goes slower. It just goes slower, but it still works. Okay, because if you cool an enzyme down, it won't break it. It'll still work, but it's not going to go as fast. So in order of speed, how fast things go, the room temperature is number 1 in speed. Then if you make it cool it down, it'll be a little slower, number 2. And hot is doesn't even count because it didn't even work. So when we changed the pH, we made it more acidic or basic. It changed how well the enzyme worked. Remember, An acid and a base are like on this little scale here. We have water in the middle, and we get a bunch of H pluses. We make it an acid. And if it gets down to a number 1, way down here, let's say this is number 7. Let's say this is number 4. This is going to be a 10. This is 14. This is going to be an acid that melts you. Okay? You can get an acid that melts you because you get H pluses. You get H pluses. And the more H pluses you get, the more acidic it is. Okay? So we have lots of H pluses. We have some H pluses here. Remember, water is H2O. It can turn into OH negative or H plus. So water's in the middle at pH of 7. At pH of 7, it can also become basic, and we can get some OH negatives or lots of OH negatives. And that is basic, and it's a base that will melt you. Just like acid, it will melt you. So that's pH scale in a nutshell. If you want to learn more about that, you can ask me. But basically, water can turn into something like OH negative or H plus. If we turn it into an H plus, it's going to make it more acidic and make it an acid. And if we get too many H pluses, it will melt you. And if we get too many OH negatives and make it a base, it will also melt you. So what happened to the rate of reaction? Meaning the rate of reaction is how many bubbles, how fast bubbles were made. Okay? Because we were looking at how fast the reaction happened. We were looking at how many bubbles we made. What happened to the rate of reaction when we made it more acidic or basic? So this is acidic. This is basic. Okay? And this right here is 7. This is water. It's neutral. So if you look here on this little graph, the enzyme works best. The enzyme is fastest in water. And if we turn it in a base, or we make it an acid, right here, this is a base, the enzyme will go slower, but it still works. And over here, if we make it an acid, right, this is acid, this is a base. Oops, I just showed you that. If you turn in, make it into an acid, the enzyme is going to go slower. And if you look over here, way down here, if we make it too acidic, and we're going to melt the enzyme. So enzyme is melted and broken. Okay? So here's our lab. We have pH of 7, meaning water, and we have lots of bubbles. Okay? This is HCl, which is an acid, and this is NaOH, which is a base. Okay? And there are, right here you can see only a few bubbles. So that's what happened to the enzyme. When we changed the pH, the enzyme didn't work as well, and it got slower. So we only got a few little bubbles. But when we put it at pH of 7 in water, the enzyme worked really well. So how does this prove that enzymes are affected by their environment? Well, the environment is two things, right? We did temperature and we did pH. When we changed the temperature and pH, we changed how well the enzyme worked. So when we changed the temperature or pH, right? You could put either one, blank. Temperature and pH. The enzyme worked differently. For example, when you boil the enzyme, you change the environment and break it. So the enzyme doesn't work, and makes no bubbles. Okay? And for the pH one, if you change the pH, you also get less bubbles, because the environment is affecting the enzyme. The environment is what's around the enzyme, so if we change what's around it by making it an acid or base, affect the enzyme. If you change the environment, you're going to change how well the enzyme works. So that proves that enzymes are affected by their environment, because when we change the environment, how well the enzyme worked changed. Cells. So let's start reviewing what cells are. What are the differences between plant and animal cells? Plant and animal cells, let me label these for you. This is an animal cell, and this is a plant cell. A plant cell has all of the things that an animal cell does, right? We've got the nucleus, we've got the endoplasmic reticulum, the Golgi apparatus. They have the same thing. You can see it there. We've both got the mitochondria. We've both got these little green things, which are lysosomes, or vesicles. They have all the same things, but a plant has extra special things. It has the vacuole. It has the chloroplast. And it has the cell wall. It has three things an animal does not have. It has a vacuole for storage, a chloroplast for photosynthesis, and a cell wall to make it sturdy. Right? A cell wall makes the plant really hard. Those are three things that a plant cell has that an animal doesn't. So an animal cell is flexible, and the plant cell is not flexible because of the cell wall. Animal cells need to get their food from somewhere else, but plants can make it using their chloroplast and doing photosynthesis. Animal cells don't have a storage site, but plant cells have a vacuole to store things like water and other stuff. And these three things are the organelles a plant has. The special organelles a plant has. Right? These are the special organelles a plant has. Vacuole, chloroplast, cell wall. What are the difference between eukaryotic and prokaryotic cells? Eukaryotic, right, is us, and prokaryotic is bacteria. The first big difference is size. Okay? Eukaryotic over here, eukaryotic, and much bigger than prokaryotic. So eukaryotic cells are way bigger than prokaryotic. Okay? Our cells are much bigger than bacteria. The first difference is size. But it's hard to see this, so I'm going to make the bacteria look bigger. It doesn't actually look that big. So, second difference, if you look into a bacteria, you'll see that there's no nucleus. This is missing. There's no nucleus. The DNA or the RNA, which is genetic material, is just floating around. There's no nucleus in the prokaryotic cells. In a prokaryotic cell, there's no nucleus. In a eukaryotic, we've got a nucleus to hold DNA. Or RNA. Well, let's make it DNA. To hold DNA. A eukaryotic cell's got a nucleus to put the DNA in. The prokaryotic doesn't have that, and the DNA is floating around. The third difference is the prokaryotic cell has no membrane bound organelles. All that means is if you look in a prokaryotic cell, there's just basically two things, right? There's these little dots and there's the DNA. There's no organelles. If you look in a eukaryotic cell, there is tons of membrane bound organelles. Remember, a membrane is just a layer or separation, and you can see that there's a little, this organelle's got a wall between it and it's a little special compartment. You've got a special space over here. You've got a special space there. We've got a special space right here. There are tons of different membrane-bound organelles that separate spaces inside a eukaryotic cell. It's nice and organized. This is it's nice and organized. Okay? Things just float, but in the prokaryotic cell, things just float around. Okay? That's difference number 3. Difference number 2 is the nucleus. And difference number 1 is size. You should talk about those 3 things. 1, 2, 3. Draw and label a plant cell in the circle below. Then add all the parts of the cell listed below. The circle is one part of the cell. Oops. The circle is one part of the cell. Alright, so in order to do this, let's take a look at what a plant cell is and remember the parts. The nucleus is that big central thing. The DNA is inside of it. The endoplasmic reticulum is these blue things right there. The Golgi apparatus is over here. Ribosomes are the little red dots on the endoplasmic reticulum. Lysosomes are these little circles right here. They help digest things. The centrioles are hiding actually in this picture. You can't really see them. So we're going to have to add them. This is the centrioles right here. Anyways, so centrioles will be right there. The cytoskeleton, I'm going to have to go all the way around here. It's right there. It's those yellow things. You can see the cytoskeleton. It's twisted through and around the cell. The mitochondria is right here. And the chloroplast is right here. And I'm going to erase these so that I can start over. The vacuole is over here. Plasma membrane is the wall. The yellow inside membrane. And the cell wall is the outside one. There's two layers there, right? There's one on the inside, the super skinny thin one. That's the cell membrane. The cell wall is the big fat thick one on the outside, okay? Those are all the parts of the cell. You can go back and review that if you'd like. So, I need to draw that. Alright, so let's see. I'm going to start by drawing my nucleus. So I'm going to start with the nucleus. I'm going to draw a circle, and there's my nucleus. Nucleus. And I'm going to put some DNA inside of it. DNA is inside the nucleus. Now I need an endoplasmic reticulum. Let's see. It's going to go back and forth, and it's right around the nucleus. Okay? It's right around it like this. This is my endoplasmic reticulum. You should show it right outside your nucleus, okay? The Golgi apparatus I'm going to put over here. It's also a membrane that goes back and forth like this, right? This is my Golgi apparatus. I also need some ribosomes. Well, if you remember, ribosomes should be on the endoplasmic reticulum, because they're right there. Those are ribosomes. Ribosomes. Now I need some lysosomes. Oh, those are easy. I can just draw a few little circles right here. These are lysosomes. Okay? Those purple circles. Centrioles. Let's do orange. Centrioles, they're hard to draw, but I can just do these circles, these little tubes, right? So there's one tube, and there is another tube. Those are the centrioles. I should show it as a tube, because that tube should have the cytoskeleton coming out of it, right? The cytoskeleton, right? This is the cytoskeleton. The cytoskeleton should be coming out of the centrioles. Then you've got the mitochondria. The mitochondria, you should draw it looking like a little bean. And then you should have this little, the little back and forth thing going on inside of it. This is the mitochondria. And the chloroplast, you can draw as a nice big circle like this, and draw all those little stacks inside of it. Right? Those green little stacks. This is going to be the chloroplast. I also need a vacuole. Oh, that's easy. I'm just going to draw, I am just going to draw a nice big little big bubble here. There's a vacuole. Done. And last but not least, I need a plasma membrane and a cell wall. Well, that's easy. I just need to draw another line here. So I'm going to draw a big, I'm going to make this my cell wall on the outside. So this right here is going to be my cell wall, this big thick black thing. And it goes all the way around the cell. Okay, so I can label that. That's the cell wall. And it's super thick, right? If I was going to color it in, it's super thick. But on the inside, I've got the plasma membrane. So I'm making the circle my plasma membrane. The little inside circle right here. Oops. I erased it. The little inside circle right here is my plasma membrane. The outside layer is the cell wall, okay? So the plasma membrane is the inside circle, the cell wall is the big outside one. That's how you could draw and label a plant cell. So writing the functions in a sentence or less. So this should make this super easy on yourself, right? You don't want this to be too hard. I'm going to pick easy definitions. So the nucleus holds genetic material. DNA is genetic material. The endoplasmic reticulum, it folds proteins and makes them into those cool shapes. The the ribosomes make proteins. That's why they're on the endoplasmic reticulum. The Golgi apparatus packages and sorts things. The lysosomes break things down. Centrioles organize the cell. Cytoskeleton is the cell's skeleton. For support. The mitochondria, it makes ATP, energy for cells. That is the job of the mitochondria. The chloroplast turns light into sugar. The vacuole is for storage. The plasma membrane is a barrier, right? We talked about this, a barrier to keep things in or out, and the cell wall is to make it sturdy and tough. Makes the cell sturdy and tough. Great. Those are definitions that you could put and it would give you full credit. You don't have to make them too fancy. Cellular respiration and photosynthesis. Cellular respiration and photosynthesis notes. Let's get started. So, cellular respiration What's the big equation for cellular respiration? Well, we take C6H12O6, which is sugar, and 6 oxygens. See the 6 right there? We take 6 O2s, 6 oxygens, and we turn them into 6 carbon dioxides and 6 waters and ATP. That's what you have to memorize. We take the energy out of the sugar and we put it into ATP. So where does cellular respiration with oxygen happen in the cell? Well, after we get some sugar, we're going to take it into the cell and do some things to it, and then we're going to take it, if we have oxygen, into the mitochondria. So the answer for this is a one-word answer. It is just mitochondria is where we do cellular respiration to make ATP, because that's the job of the mitochondria, to make ATP. What's the big equation for photosynthesis? We take 6 CO2s, so 6 carbon dioxides, and we take 6 waters, and we take light energy and we have a reaction, and we stick the other light energy into glucose, which is a sugar, and 6 oxygens is something else that we pull out. We put all the energy from light into the sugar, and we get some oxygen as a waste product. So we take 6 carbon dioxides and 6 waters and add some light, air and water. Then we turn that into glucose, sugar, and oxygens. You have to memorize that. Where does photosynthesis happen in a plant cell? The chloroplast is where photosynthesis happens, where light is going to go in there and get turned into sugar. It happens inside the chloroplast. Photosynthesis lab review. So, in the photosynthesis lab, we took leaf discs and put them in water with some baking soda. So we took some baking soda, right? And we took some leaf discs, right? And then we put them in the sun. So we had a bunch of leaf discs, and we had sunk them. We made them sink. So we took sunken leaf discs, because normally leaf discs float, but we made them sink. Right here, we used the syringe. We used the syringe to pull water. Pull air out of the leaves to make them sink. Okay? So we had all of these sunken leaf discs sitting on the bottom of our cup. And then we put them in the sun. And the leaf discs floated. Ta-da! They all started to float, and they worked their little way up to the very top of the water when we put them in the sun. Now, why did that happen? Well, we took the energy from the sun, you know, that light energy, and that was light, right? We put that into the cells, and they did photosynthesis. So the leaves, it's actually the chloroplasts. The chloroplasts took the light, the CO2, carbon dioxide, and some water, and did photosynthesis. And they made oxygen. And if you have all these little air bubbles in your leaf, you're going to make it float. Okay? So we made air. The air, right, which is oxygen, the oxygen the leaf discs made them float. Okay? Normally, we have no air in here. Right in here, at first, there is no air. But after we stick them in the sun and take that light and do photosynthesis, we make some air, and all of a sudden the leaf discs start to float. Up to the very top of the cup. So if we put the leaf discs in the dark, they would eventually sink again. What is happening that makes them sink? Well, we've got all of this oxygen here, right? And we don't have any more life, so we're not going to make any more. But what we are going to do is start using the energy we made as a plant. You know, a plant needs to eat too. A plant also does cellular respiration. So we're going to use the oxygen. The plant uses the oxygen to do cellular respiration. Plants have mitochondria and they do cellular respiration too. The plant uses the oxygen to do cellular respiration, and all the air, all the oxygen gets taken out. And when that happens, the leaf discs are going to sink again. So we take that air that we made through photosynthesis and we use it to do cellular respiration. And then the leaf discs are going to sink back down to the bottom of the cup. Normally, we do so much photosynthesis that the leaf discs float, and they don't sink, even though cellular respiration is happening. But when we stop the photosynthesis, cellular respiration can catch up and use up all of the oxygen. Okay. Macromolecules. Macromolecules. So, what are the four types of macromolecules? Well, there are carbohydrates, there are proteins, there are nucleic acids, and there are lipids. And remember, these are macromolecules. They are big molecules that make up who we are. The macromolecules are the four basic building blocks of life. Okay? What are the functions of each macromolecule? Well, carbohydrates are for fuel and building material. We get energy out of them, and we can use them to do some building. We do that in plants. Proteins are for structural support, storage, transport, cellular communications, movement, and defense against foreign substances. And that is a ton of stuff that they do. So you can say, if you want to on the test, you could say pretty much everything, like muscles and enzymes, though, to give an example of two things they do. Lipids are used for long-term energy, membranes, and hormones. And nucleic acid are used to make the genetic code right? DNA. Those are the functions of each one. So how do you make a macromolecule? What's the reaction you do? It's called a dehydration reaction, where you take out water to make a new bond. Okay? That's what you would say. You would say you do a dehydration reaction where you take out a water to stick to things together. That's what you do. How do you break a macromolecule down? You do hydrolysis, where you add a water to split two things apart. That's how you break a macromolecule down. And we do this all the time in our bodies. So, macromolecules lab review. In the macromolecules lab, we tested for different macromolecules in foods. What type of macromolecule was each thing? Well, glucose and dextrose, if you look, it's a carbohydrate, right? It's a sugar. It's for energy. Starch is also a carbohydrate. It's a carb. They're carbohydrates, but they're different types. Protein powder is a protein, and oil is a lipid. Okay? How could we use the tests for macromolecules to find the thief? Well, we did 4 different tests. We did a glucose, a starch, a protein, and a lipid test. And each test, we could find out what was inside a food. So in the glucose test, we found out if there was glucose because it turned orange. In the starch test, we found out if there was starch because it turned this dark purple. In the protein test, it turned purple if it has proteins. And in the lipid test, the fat, the lipids, would leave a stain on the paper that you could see light through. So we used that to do this testing. We filled out this massive table, right? We had the evidence of who the thief was, and our job was to match that evidence to the type of food it was. So we did all these different tests, and we found out what was in each type of food. For example, in pretzels, we found out, yeah, pretzels have sugar and some starch. They're made of flour. No, pretzels don't really have protein, and they're not full of fat. Okay. And so if we looked at the evidence down here, we'd say, oh yeah, well, the evidence has sugar too, but the evidence isn't made with flour, so that means it can't have been the person who was eating pretzels. So in the end, if you figure this out, you look at the yogurt or the milk, and it has sugar, no flour, and it has proteins. And that's the same as the evidence. So Ashley has to be the thief because she's the person who's eating the fat-free yogurt. And she is the thief because we can match the evidence she left at the scene of the crime when she stole the iPod, with testing other milk. We tested some other milk or yogurt, and we found out that it matched the evidence left at the scene of the crime. So we found out that she was the thief. So how did the lab tests show you that all the things in plants and animals are made from the same 4 types of macromolecules? Well, here's the deal. Did you ever get a food that didn't have any of the macromolecules? If you think about it, you realize that every single thing you tested, except for water, which isn't a macromolecule, every single thing you tested positive in at least one of the tests. It has something inside of it. All the things that come from a plant or an animal that are food, which basically we get out of a plant or animal, have the macromolecules. They all will have some macromolecule inside them and test positive in a test. The other question is, did foods from an animal and a plant have the same macromolecules? Well, if you remember, beans come from plants and cheese come from animals. And when we tested those, we found out that, you know what, cheese and beans have the same test results. A plant and an animal, a food from a plant, beans, and one from an animal, cheese, have the same macromolecules and test positive in the same tests. So we know that all the same macromolecules, that the same 4 types of macromolecules, we can find those in every single thing that's in a plant or animal, because they're all we use the same macromolecules in plants and animals and everything on Earth. We all use the same ones because they all will test positive in the tests. So how did the lab test show you that the 4 macromolecules have different properties? Well, we had to use 4 different tests. The glucose test only reacted with this special molecule right here, right? It wouldn't touch anything else. This is the only one that would turn orange. The starch test is the only thing it would react with the starch, and then it would turn purple. The protein would only turn purple if there was protein there. And the lipids would only leave a stain if it has fats inside of it. So each of the macromolecules have different properties, and we can use those properties to help us test to find them. Because if they were all the same, we couldn't tell them apart. But because they're different, we can use different tests on them. How did the lab test show you that polymers are different than their monomers? Polymers, remember, are long chains of molecules, and monomers are single pieces. Well, let's take a look at the glucose tests and the starch tests. We had carbohydrates, right? Glucose and starch are both carbohydrates. Glucose is the monomer of starch. I take a piece of glucose and I stick those together in a long chain. I make a long chain of glucoses, and I will make a starch molecule. It's made of the same thing. A starch is just a lot of glucoses. But the starch won't show up in the glucose test. It won't do anything. When I put the starch in the glucose test, it stays blue. This would be the starch right here. It would be blue if I did the glucose test because there's no glucose. The test won't find anything. And if I do this, put the glucose in the starch test, this would be the glucose right here. It would still be orange because the iodine won't react with it. If I put a bunch of glucoses together, it's going to be a different thing and have different properties. And I'm going to have to use a different test to find it. So I'm going to have to use a starch test if I put a bunch of glucoses together, because I now have a polymer of glucose. And a polymer of glucose has different properties, and I need to use a different test. As same as the monomer of starch has different properties than starch, and I have to use this different glucose test to see where glucose is.